I want to talk to you today about this problem of natural evil. And this was a challenge that a colleague posed to me over 20 years ago when we were talking about the flagellum as being irreducibly complex. If you know some of Mike Behe's work, these organelles that are exquisite nanomachines, and as evidence that there is, design in nature, true design, not just apparent design, but true design. These are complex molecular nanomachines that really outstrip any contrivance that we have been able to build. But the challenge was, if you think God created the flagellum, did he create the type 3 secretion system, something else that we worked on, which I'll explain today, which is probably the most powerful weapon bacteria use to cause sickness on this planet. So, and it's irreducibly complex too. So did God create this as well? And if he did, then is he not the author of evil? This is an old question, and it goes all the way back to a famous letter. Well, let me start with this in terms of defining what we're talking about. When I was contemplating this question 20 years ago, I started rereading Mere Christianity by C.S. Lewis. I probably read this about 8 times in my life. And in chapter 5, he talks about spiritual evil, and he makes a statement that a good world that has gone wrong, but still recalls a memory of what it ought to have been. In other words, we look at the world today, we know what perfection should be, but we don't see it. But we have this nostalgia in terms of what things should be like. It is the real recognition of the fact that evil is a parasite, not an original thing. So he's talking about spiritual evil, but that really sparked my interest. Could this be true for natural evil as well? We know that parasites are dependent upon a host, right? And natural evil, could it be that evil is always derivative of good? It's always a perversion of good. And we know this, that you can have good without evil, but you can't have evil without good. Evil is dependent, it's a derivative of good. And think about that as you grow, because I think that's a key observation. So I started thinking, do bacterial pathogens that I work on, have they evolved from a harmless progenitor, a harmless antecedent? And could the loss of genes contribute to virulence? So loss of information. This was a radical idea, 15 years ago in bacterial pathogenicity. And it started to kind of foment as we were working. Now here's Darwin's perspective. And again, I run into this all the time on the university, and you will run into it as well. And it may even derail you if you haven't done your homework. But this is a famous letter to Asa Gray that Darwin wrote in May of 1860. He said, "This is always painful to me. I am bewildered. I had no intention to write atheistically, but I own that I cannot see as plainly as others do, and as I should wish to do, evidence of design and beneficence on all sides of us. There seems to be too much misery in the world. I cannot persuade myself that a beneficent, omnipotent God would have designedly created the Ecumenidae, these are parasitic wasps up here, with the express intention of their feeding within the living bodies of caterpillars." So these Ecumenidae wasps, these parasitic wasps, you see this one here, is depositing its eggs in this caterpillar. He's got an ovipositor or a stinger. He injects eggs into that caterpillar, and those eggs hatch and eat the caterpillar from the inside out. This looks pretty bad, right? Did God create this? It's a good question. Again, I don't know if you can see this, but these are all little larvae that have hatched from eggs within that poor caterpillar and use it really as their food source. So natural evil, did God create these parasitic wasps for this purpose? And then, as I mentioned, I hear this all the time, and here's actually a really good conversation between Stephen Meyer and a couple of mathematicians about, evolution in general. But this question of evil comes up. And both of the scientists that Stephen Meyer is talking with come to the conclusion, because there's so much evil in the world, that God failed. It would have been better if he hadn't created the world. And Steve comes back with some of the arguments actually that we've talked about together. But in my view, forget about parasitic wasps. That's nothing. Let's talk about the organism I work with, Yersinia pestis, which causes bubonic plague. There's an electron micrograph of the surface of the cell. I don't know if you can see them, but these arrows are pointing to these little hair-like projections from the surface. These are nanosyringes, right? Just like the parasitic wasp injects, eggs into a caterpillar. This bacterium, if you are so unfortunate enough to catch it, will use these little stingers to inject your cells directly with proteins that shut down your immune system. This is bubonic plague, all right? When we blow up these little structures, this is the outer membrane of the cell, this is the inner membrane. These are little nanosyringes. Here's a cartoon, so we know all the parts. And the plague has had a profound effect has had a profound effect on human history. We've had 3 pandemics, which should be a word familiar to you after the last few years, of plague in the world. The first one recorded was the Justinian plague, when Justus was emperor of Rome, back in the 6th century. We're looking here at the number of years that epidemic occurred or lasted, or pandemic lasted. And here we're measuring the number of deaths that we estimated occurred in millions. So this is 50 million up at the top, and this is, you could calculate the area under the curve. That's the first recorded pandemic. The Black Death that occurred in the 14th century between 1347 and 52, we estimate killed a quarter to a third of the population of Europe in 5 years. And there were aftershocks of that occurred every 50 to 100 years. And we still have cases of plague in the United States. So the third pandemic occurred at the turn of the last century, the late 1800s. And as I said, we still have outbreaks today. We had a case of plague in Idaho about 4 years ago down in Jerome County. Overall, we estimate plague has been responsible for 300,000 deaths. All right? So again, forget about Darwin and stinging wasps. This is an important question to address. Here's the spread of the Black Death in the 14th century. Came from the steppes of Russia along the trade routes, and here you can see the years 1347 in color to 1352 as it rapidly spread back then when all you had were ships and land routes for trade and transmission. A frightening thing. Imagine if you look back in this time period, and in the course of a week, 60 to 80% of the people in your town in Moscow are dead, right? It was devastating in terms of its impact economically, and it took over 200 years really to recover from that plague. This is a cartoon showing that plague exists in really 2 cycles. We talk about sylvatic, and you're taking Latin. Sylvatic literally means of the woods. So it's primarily what we call a zoonotic disease. Man is an incidental host. Rodents are the primary hosts for plague. They can go to carnivores if they eat these rodents, but rats are the main reservoir, and it's transmitted from rat to rat by fleas. So this bacterium has adjusted or adapted to grow within a flea and a mammalian host, very different environments. If these fleas bite you, they can transmit the organism into your system. We have urban plague where it gets into the urban rats, and this is what occurred in Europe in these other pandemics that have occurred. And as the rats die, the fleas will seek an alternative host to feed on, which is us. Okay, missed one. Okay. Oh, there we go. So this bacterium, I don't want to go into too much detail, but it's classed in what we call gram-negative bacteria. They have a certain surface structure. I don't know if you've encountered those, but they're very much like Escherichia coli. Sorry.
They're standing to say hi to a board member who just came in, Sam.
Okay.
Thanks, guys.
But Yersinia pestis is the name of the plague organism. The name is derived from Alexander Yersin, who was one of Pasteur's students that isolated it and discovered it in late 1800s in China. But it's a gram-negative bacteria, very similar to E. coli, Escherichia coli, which is our workhorse in molecular biology, and Salmonella. The mortality rate, if untreated, is around 50 to 80%. So if you contract this disease, 50 to 80% of the time you're going to die, right? It causes it's the most virulent bacterium that we know of, and it causes 3 syndromes of disease: bubonic, septicemic, which means it goes directly into the blood and you're dead usually within 24 hours, and pneumonic, where the septicemic gets into your lungs, and then when you cough, you can spread it directly to someone else in close contact. You bypass the need for a flea to transmit the disease. There are 2 other closely related species that cause human disease: Yersinia pseudotuberculosis and Yersinia enterocollitica. These cause a gastroenteritis. You acquire them by eating contaminated food or water. They cause a self-limiting disease. Usually we don't even treat them, but it's interesting that at the DNA level, Yersinia pseudotuberculosis is 99% identical to Yersinia pestis. So a fairly innocuous organism is first cousins to the most pathogenic bacterium we know of. Here's what bubonic plague looks like. The bubonic comes from the fact that you get these buboes. You get bitten by the flea, the organisms migrate through the lymph system to the closest lymph node, and they set up shop, and you get this swelling, localized swelling. Eventually this will burst and spread throughout your system, and you're, like I said, dead usually within 24 to 48 hours. So these, you get them wherever the flea bites you, usually in the neck, the groin, under the armpit. Cause of Black Death for a reason. This is a Chinese patient that's recovering, but you go into severe hypotension, your blood pressure drops, you lose blood flow to your extremities. They can undergo necrosis, and as you see, this patient has lost some toes, although he's recovering. Says that at one point his entire body was almost black from the severe effects of the disease. Pretty nasty, right? And again, we're asking questions. Where'd this organism come from? So here's a schematic where we have a bacterial cell, and I've divided it in the middle. On this side, we have Yersinia enterocollitica, or Yersinia pseudotuberculosis, fairly innocuous. And on this side is plague. So again, these are very similar organisms. The genome size is like E. coli, about 4.3 million base pairs of DNA, AGCTs, 4.3 million of them in a big circle. That's the code. But they also have these extra chromosomal DNA elements we call plasmids. And if you're going to cause disease in your Yersinia, you have to have this plasmid that's present in both of them. This codes for the type 3 needles, those nanosyringes. This is not part of the original chromosome. This was acquired by what we call lateral gene transfer from some other source. All right, so if you cure either one of these organisms of this plasmid, you can eat it, it won't hurt you. You can inject it into your blood and you'll clear it immediately. Yersinia pestis has acquired 2 other plasmids, one allowing it to grow in the flea, another one allowing it to codes for this phospholipase D, which allows survival in the flea, and trying to see here, Pla protease. This protein is essential for it to get out of the lymph nodes and spread throughout your body. All right, that's encoded on the small plasmid. All of these smaller pieces of DNA were acquired by lateral gene transfer. They weren't part of the original genome of the organism. That's an important fact, I think. All right? And these organisms are modal. They make a flagellum. They can swim. Yersinia pestis is non-modal. But students in my lab showed that all of the genes for making a flagellum are on the chromosome of the plague organism. They've lost function. I'll show you why that's important. These organisms are modal at 30 degrees Celsius, but they lose production or synthesis of the flagellum at 37 degrees Celsius. What is that temperature? The body temperature for a mammal. So you express these flagella at 30 degrees, but if you hit 37 degrees, which is a mammalian host, a human or a mouse, you shut off the flagellum. Again, that's very important. It's temperature sensitive. So we started out just asking, how does temperature regulate gene expression? Very basic question and very fascinating, but we're not going to go into it. All right, again, Yersinia pseudotuberculosis intercolitica. They cause localized infection in the gut. You eat contaminated food or water. They set up shop in the gut. They invade through these antigen sampling cells in the gut lining, so these are your microvilli, and these M cells, and underneath are macrophages, white blood cells that are our primary defense system against an invader. So they kind of head for the Pentagon where they want to set up shop. But eventually they're cleared, and as I said, they're localized, self-limiting. Usually we don't even treat them with antibiotics. On the contrary, here's plague. You can get systemic infection. It can go all over, get into your lungs, and you can spread it directly. So here's a schematic of what we think. Now we have excellent evidence that this is what occurred. You started out with some non-pathogenic gram-negative bacterium, and at some point, by lateral gene transfer, it acquired that plasmid that codes for those nanosyringes. Now it can cause disease. The syringe injects proteins that shut down your primary immune system. There's no inflammatory response. Right? That's critical for bacterium if they're so unfortunate to catch a mammalian host. Because we're going to throw everything at them. They're going to get phagocytized, be in a vacuole. We're going to fuse that vacuole with things that produce nitrous oxide, bleach, high temperature, proteases, and dissolve that invader so there's nothing left but toenails and earlobes. But if you have this, you can escape that process. And then it acquires some other genes on the chromosome by lateral gene transfer that allow it to form biofilms and grow in fleas. Another plasmid was acquired that makes a capsule that can also help evade the immune system. You kind of hide your surface in a protein coat that's not recognized. You got the small plasmid that was acquired that codes for this claw protease. And so most of these virulence or weapon systems have been acquired by some other source, and they're being expressed out of context. All right? I'll come back to that. But that to me is, I think, an important clue and very satisfying. But that's not enough, because we can take Yersinia pseudotuberculosis and put these other two plasmids in it, and it doesn't cause bubonic plague, right? And there's nothing else on the chromosome that's different from the plague organism. So the question was, what is it? There aren't any other weapon systems that we can identify. So going back to comparing these organisms, pseudotuberculosis is 99% identical to Yersinia pestis on the chromosome. But when we finally got the entire genomic sequences to compare to the nucleotide, 4.3 million base pairs, what's different? We found that, surprisingly, 13% of the genome of Yersinia pestis has been inactivated by mutations. Right? That's interesting. You might expect that because it's a parasite, and maybe it's depending upon its new host, whether it's a flea or the mammalian, to provide amino acids or vitamins. It doesn't have to make them anymore, so those you lose by atrophy. But at this point, we proposed in my lab that this may be essential for virulence. As we're beginning to understand more about the immune system, we know that some components of the bacterium are important as recognition signals for our immune system. Okay, this was published in 2004 in the Proceedings of the National Academy of Science, and looking at these results, authors said these results provide a sobering example of how a highly virulent epidemic clone can suddenly emerge from a less virulent, closely related species. So I had a medical student in the mid-1990s that we're looking at flagellum regulation and temperature regulation of flagellum biosynthesis. We speculated that plague might have been modal at some point. We cloned the genes, and this student sequenced this gene we call FLHD. This is a key master control governing 50 genes to make the flagellum, so this organism can swim. Yersinia pestis has never been observed to swim, and he found, comparing these, so this is a sequence of Yersinia pseudotuberculosis, right? Read in triplets, ATG is the first codon in that protein, stands for methionine, will code for methionine, an amino acid. And down here, there's a single T insertion. We call that a frame shift mutation. Suddenly, you throw off the register of nucleotides as they're read, and you start inserting the wrong amino acid. And eventually, you make a non-functional protein. That was in '94, before we had the genomes. Now we have the genomes of all the organisms around the world. And when we compare them, every single one of them, whether they're from China, from the steppes of Russia, from the southeast or southwest United States, they all have the same single nucleotide insertion. That means that this mutation occurred very early in the evolution of this pathogen. Why would that be important? We also know that the flagellum, shown here, you have your basal body, this is your U-joint, and the filament protein will be attached to this. It's very similar to these needle-like structures. And in fact, from experiments in my lab, we showed that if you make the filament protein of a flagellum artificially at 37 degrees, in Yersinia intercolitica, you're not modal, but that flagellum will be recognized and exported out, injected into host cells using that type 3 secretion system. They have reciprocal recognition. They're very specific protein secretory machines, and they will cross-recognize the substrates used. You can take one of the effector proteins that shuts down your immune system and express it at low temperature, and it will be exported out to flagellum. Now we come to the crux. Why is it important to lose flagellum biosynthesis or shut it down if you're a gram-negative pathogen? The filament protein, right, the propeller of the flagellum, there's about 50,000 proteins in this that assemble into this long polymer. We have specific receptors within our cells and on the surface of our cells that are looking for this pattern recognition molecule. You only find flagella in bacteria, and we have specific receptors, toll-like receptor 5 or NOD protein intracellularly. And here's our needle syringe. So if this bacterium runs into a eukaryotic, one of our white blood cells, it's going to inject flagellum, if it's made, directly into the cells that are looking for it to turn on the innate immune response. So if you want to be a successful pathogen, you don't show that protein. You shut it off if you're Yersinia tuberculosis, or because insects have these same receptors, you can't afford to express it at low temperature in plague, and you lose that whole capability of motility. Any questions on this? I think this is really key. So we can repair that mutation that I showed you, that single base pair mutation. I'm going to come back to this slide. Maybe I didn't include it.
Sam, do you mind retouching on the importance of the similarities and differences between the flagellum and the needle mechanism in the previous slide?
So they're both these organelles that are assembled on the surface. And the Yersinia only make them at 37 degrees. They're expensive to make. And if you're living in the environment, you know, if you're a facultative pathogen, you can live inside a host or you can make your living, you know, out in the environment. It's an advantage to have a flagellum, right? They're both dedicated secretory machines. So this protein, blue, which assembles into this filament, is actually made in the cell, is transported to the basal body of the flagellum, out this hollow core, and assembles at the distal tip. Very different from your hairs, which, you know, you add hair proteins from the surface of your head and push your hairs out. These proteins are exported out the hollow core of the flagellum where they assemble on the tip. Right? But this structure is very similar, and then it's dedicated to secreting proteins, but these are anti-host factors. And the point being that the recognition system for proteins to be exported by the flagellum or the type 3 secretion system is the same. They have specific zip codes built into those proteins that direct them to the site where they get used to be pushed out of the cell. Right? So a select group of proteins. Did that answer your question? All right. What's interesting is we now have almost a complete history of the evolution of plague, and it's occurred very recently. These skeletons that were these burial grounds found in Central Europe and on the steppes of Russia from the Bronze Age or even a little bit earlier were exhumed, and people isolated DNA from these skeletons from the teeth pulp, wanting to know what the origin was. We compare the genomic sequences of human DNA to see what the lineage of these people were back in the Bronze Age. And it found that 7 of skeletons of 100 that were exhumed also had died of plague. We reassembled the entire genome of Yersinia pestis from the multiple sites. And what I find fascinating is that this is a time scale. This is looking at the lineage of plague about 3700 BC. The mutation that we identified here at the University of Idaho, that single T insertion shows up in these fossilized records. All right, and that, like I said, we can repair that mutation, and the organisms can't cause disease because our immune system picks it up. This was published a few years ago by colleagues in Cambridge. But in summary, Yersinia pestis is a recent clone, less than 6,000 years ago, of Yersinia tuberculosis. Virulence is due to both acquisition of genes, bilateral gene transfer, and gene loss. We now know there's another single nucleotide mutation that's involved in the assembly of the outer membrane of these bacteria that modify it at 37 degrees, allowing, because there's a receptor looking for that lipid molecule that's specific for bacteria, to set off our immune system. And we can repair that, and again, Yersinia pestis is innocuous. 2 single nucleotide changes, loss of function, allow this organism to cause devastating disease. So this most virulent bacterium arose by expressing genes either out of context, they acquire them by lateral gene transfer, or gene loss. You lose motility, you lose modification of your outer membrane, and you can come under the radar of your primary defense system that's looking for these cellular molecules. It's warm in here, right? You guys are just ready to pass out.
Open that window. Lizzie, mind
the
opening?
important thing I want you to take home message, you know, when you run into these arguments, did God create AIDS? Did God create parasitic wasp? Did God create the plague? We can say definitively that the original design, the aboriginal design of plague was not virulent. This is through degradation of information, loss of information, or expressing information out of context. And I find that very satisfying. You know, after 20 years of mulling over this and thinking about it, and getting a hint out of C.S. Lewis' book that was published back in the 40s. It's true. Now we come back full circle to Darwin's problem with these parasitic wasps. There's a paper published in 2009 in the Journal of Invertebrate Pathology, and as it turns out, early in the history of these parasitic wasps, they were infected with a poly, where is it? Remarkable exception. Poly DNA viruses or poly. They were infected with an insect virus, and part of the virus genome was incorporated into the insect genome. There's lateral gene transfer. Those genes are only expressed in ovary tissue when these wasps are ovulating, and those proteins are injected with the eggs into the caterpillar, and they shut down the immune system, the primary immune system of the caterpillar, so they don't recognize they've been invaded. Almost the same scenario. You could say that these parasitic wasps, at one point in their history, were not designed for this. It was by acquisition of lateral gene transfer by a virus that gave them this capability. So, from our perspective, I find it satisfying because it fits with our idea of the fall, and that when evil was introduced into our environment, that there was this degradation of information, or trading of information, that allowed these pathogens to cause misery. But it wasn't the original design. All right? So, these examples of natural evil and parasites show that malevolence was not part of the aboriginal or original design. So I'll stop there and